Good morning, everyone, and welcome. I'm glad to see so many of you here today. I am Dr. S.K. Singh, holding a PhD, and I will be guiding you through this presentation. Today, we will be discussing quality control procedures in liquid manufacturing and packaging. Over the next few minutes, we'll explore key aspects of ensuring quality at each stage of the process. So, let's get started and dive into the important practices that help us maintain high standards in our industry. Let's start by discussing the main objectives of quality control, or QC. The primary goal is to ensure that our products are consistent and safe for use. This means verifying their identity, making sure they are pure, have the correct strength, and remain stable over time. By doing this, we can confidently confirm that the product meets the required standards before it reaches the customer. Another key aspect is detecting defects early on. Identifying any issues before the product is released helps us prevent problems in the market, saving time and resources, and most importantly, protecting patient safety. Additionally, QC aims to prevent contamination, whether microbial, which can cause infections, or particulates that could compromise the product's quality. Now, turning to the regulatory framework that guides our QC activities. For pharmaceuticals, we adhere to the FDA's 21 CFR Parts 210 and 211, which outline current good manufacturing practices, or CGMP. These regulations set the standards for manufacturing, processing, packaging, and holding drug products to ensure quality and safety. We also follow international guidelines, such as ICH Q7, Q8, Q9, and Q10, which cover quality principles, risk management, and process validation. The WHO GMP standards are another cornerstone, providing global best practices for pharmaceutical manufacturing. Furthermore, we rely on official compendia like USB, EP, and IP monographs, which specify detailed QC tests and acceptance criteria for various substances. To achieve all this, our QC system is built on 4 key pillars: establishing clear specifications, performing validation of processes and methods, maintaining thorough documentation, and implementing rigorous controls throughout production. These pillars work together to ensure that quality is built into every step, supporting the overall goal of delivering safe, effective medicines. Today, we're going to take a closer look at the overview of dosage forms. This slide is all about understanding the different ways medications can be formulated and delivered to patients. It's important because the choice of dosage form impacts how effectively a drug works, how easy it is to administer, and how well patients adhere to their treatment. When we talk about dosage forms, we're referring to the physical form in which a medication is produced, like tablets, capsules, liquids, or even patches. Each form has its unique advantages and challenges, depending on factors like the drug's properties, the desired speed of absorption, and patient convenience. By understanding these forms thoroughly, we can better appreciate how pharmaceutical technology plays a role in improving health outcomes. So, as we move forward, keep in mind how these different forms tie into the broader picture of drug development and patient care. Let's talk about raw material quality control. The main goal here is to make sure that the active pharmaceutical ingredients, or APIs, and excipients meet all the necessary acceptance standards before they are used in manufacturing. This is a crucial step to ensure the safety and effectiveness of the final product. Now, to achieve this, we perform a series of QC tests. First, there's the identity test, which often involves techniques like IR or FTIR spectroscopy, UV, or HPLC. These tests confirm that the material is what it's supposed to be. Next, we look at assay and purity, typically using HPLC or UV methods, to ensure the material's content is within pharmacopoeial limits. This helps verify the correct dosage and purity of the raw materials. For liquids and suspending agents, we also check the pH and solubility. These parameters are critical because they influence stability and bioavailability. And for non-sterile raw materials, microbial limits are essential. We need to make sure they are free from harmful microorganisms that could compromise safety. For example, take sorbitol solution used in syrup. It must have at least 98% purity and also meet microbial limits before it can be used. This example highlights how strict quality control ensures only suitable materials enter the manufacturing process, ultimately contributing to a safe and effective medication. Let's take a closer look at water and solvent control. First, there are different types of water we work with. Purified water, or PW, is primarily used for oral solutions and equipment washing. It's essential that this water meets strict purity standards to ensure safety and efficacy. Then, we have water for injection, or WFI, which is used specifically for injectable liquids. Because these are administered directly into the body, WFI must be of the highest purity. Moving on to quality control checks, several key parameters are monitored. Conductivity and total organic carbon, or TOC, are measured to assess the purity of the water. Low conductivity indicates minimal ionic contamination, and low TOC means fewer organic impurities. For WFI, endotoxin levels are critically important, since endotoxins can cause severe reactions if present in injectable products. Typically, the endotoxin limit is set at 0.25 EU per milliliter. Additionally, microbial monitoring is carried out regularly to prevent microbial growth, which could compromise the safety of the water and, ultimately, the product. These controls are vital because they ensure that the water used in our processes remains within the specified limits, safeguarding the quality of the final pharmaceutical products. Maintaining these standards is a fundamental part of our overall quality assurance and compliance efforts. Now, let's talk about in-process controls, or IPCs, which are crucial during liquid and suspension production. These controls help us monitor and ensure quality throughout the manufacturing process. First, we look at mixing speeds and timing. Getting the right speed and duration ensures that ingredients blend properly without overmixing or undermixing. Next, temperature control is vital, especially when working with heat-sensitive materials, because even slight temperature variations can affect product stability. We also perform pH checks regularly to maintain the correct acidity or alkalinity, which can impact product efficacy and safety. Additionally, weighing and volume checks are done to verify that the correct proportions of ingredients are used, maintaining consistency batch to batch. Moving on to real-time quality control, or QC. Here, we focus on immediate, ongoing checks during production. For example, the uniformity of mixing is assessed both visually and through sampling to ensure the mixture is consistent throughout. When dealing with suspensions, particle size becomes important, as it affects suspension stability and bioavailability. We measure particle size to ensure it meets specifications. A good example of real-time QC is suspension resuspendability. We test whether a suspension can be resuspended within a defined time after shaking. This helps us confirm that the suspension remains stable and easy to redistribute, which is essential for product performance and patient compliance. All these controls, both IPCs and real-time QC, are interconnected. They help us catch issues early, maintain product quality, and ultimately ensure we deliver safe and effective medicines. Now, let's talk about viscosity and particle size control, and why these factors really matter. First off, they directly influence how a product flows, how easy it is to pour, and how uniform the final mixture turns out. If these parameters aren't properly controlled, we might end up with issues like inconsistent dosing or poor product stability. When it comes to quality control, there are a couple of key parameters we focus on. One is viscosity, which we measure using a Brookfield viscometer. This helps us ensure the product isn't too thick or too runny. The other is particle size distribution, which we evaluate using methods like sieve analysis or laser diffraction. For example, in an oral suspension, the particle size must be less than or equal to a certain value, say X micrometers, to prevent particles from settling too quickly. This is crucial because settling can lead to dose inconsistency, which impacts patient safety and efficacy. So, controlling viscosity and particle size isn't just about meeting specifications. It's about ensuring the product performs reliably and delivers the right dose every time. These controls are essential parts of the overall quality assurance process. Now, let's take a closer look at the critical QC checks involved in filtration and sterility controls for injectables. First, filter integrity is essential, and we typically verify this with a 0.22 micron sterilizing filtration. Ensuring the filter's integrity means we are effectively removing bacteria and particulates, which is crucial for patient safety. One common method we use is the bubble point test or pressure hold test before filling, which confirms the filter is functioning properly. Next, maintaining a sterile environment is vital. This is achieved through strict control of the sterile area, which is classified as grade A or B according to GMP standards. These classifications help us uphold the highest cleanliness levels during production. Air quality control is also a key aspect. We rely on HEPA filters to trap airborne particles and regularly perform particle counts to verify air cleanliness. This helps prevent contamination from the environment. On the testing side, sterility testing according to USB 71 is performed to confirm that the final product is free of microbial contamination. Additionally, endotoxin testing, using methods like LAL or BET, ensures our products are free from bacterial endotoxins, which can cause severe reactions if present. All these controls are interconnected and form a comprehensive approach to ensuring the safety and purity of our injectable products. They are fundamental to maintaining quality and compliance throughout the manufacturing process. Now let's move on to slide 9, which focuses on assay and chemical tests. Here, the key point is the importance of QC test requirements to ensure product quality. First, we have the API assay, typically performed using HPLC or UV methods. This test confirms the actual amount of the active pharmaceutical ingredient, API, in the product, making sure it meets specified standards. Next, if our formulation includes preservatives, we need to assay those as well, to verify they are present at the correct levels, especially if applicable. Lastly, we conduct degradation products and impurity profiling. This step is crucial for identifying any potentially harmful impurities or degradation products that may form over time or under certain conditions, ensuring the product remains safe and effective throughout its shelf life. For example, in the case of an oral liquid, we require that the API content is within plus or minus 5% of what's claimed on the label. This tight control helps us maintain consistency and trust in our products. Overall, these tests form the backbone of quality control, helping us verify that each batch meets rigorous standards before reaching patients. Let's take a closer look at the physical characteristics we evaluate for liquids and suspensions. First, we perform the clarity and turbidity test to ensure there are no visible particulates, especially for solutions. This step is crucial because visible particles can indicate contamination or instability. Next, we check the color and odor to confirm they meet the expected standards. Any deviation could suggest spoilage or improper formulation. We also measure the pH to verify that it falls within the specified range, as pH affects both stability and compatibility. Lastly, we assess the density or specific gravity, which helps us understand the formulation's consistency and concentration. For example, a syrup should be clear and free of visible particles, and its pH should be between 4.5 and 6, according to the specifications. These tests collectively ensure the quality, safety, and efficacy of the liquid or suspension before it reaches the patient. Now, let's turn our attention to packaging material quality control. When we talk about packaging components, we're referring to the various elements that make up the packaging system. This includes bottles, which can be made from HDPE or glass, as well as closures, caps, labels, and cartons. Each of these components plays a crucial role in maintaining the integrity and safety of the product. In terms of QC focus, there are several key areas we need to emphasize. First is material identity and compatibility. It's essential to verify that the materials used are what they are supposed to be and that they work well together without any adverse reactions. Next, barrier properties are critical for ensuring product stability. These properties help protect the product from external factors like moisture, oxygen, and light, which can compromise quality over time. Label accuracy and claim checks are also vital. Ensuring that labels are correct, legible, and truthful helps maintain regulatory compliance and consumer trust. For example, a specific case is child-resistant caps. These are tested rigorously for proper closure torque to prevent accidental opening, and also for tamper evidence, giving consumers confidence that the product hasn't been compromised. Overall, quality control in packaging is about making sure every component functions correctly, maintains product stability, and complies with safety and labeling standards, integral parts of our overall product quality assurance. Let's take a closer look at the packaging process QC. First, inline controls are essential to ensure quality at every step. We perform fill weight and volume checks to make sure each package contains the correct amount. Seal integrity is also critical, including checking cap tightness and tamper seals to prevent contamination or tampering. Label checks are conducted to verify batch numbers, expiry dates, and instructions are accurately printed. Additionally, barcode and serialization checks help us track each package through the supply chain. Moving on to defect detection, automated visual inspection plays a vital role in identifying any visible defects or inconsistencies. We also have reject mechanisms in place for underfilled or overfilled containers, ensuring only products that meet our standards reach the customer. For example, with a 100 ml oral liquid, we typically allow a weight variation within plus or minus 2% tolerance. This consistent control helps us maintain product quality and safety throughout the packaging process. Overall, these controls and detection methods are key to maintaining the integrity and reliability of our packaging. Let's take a look at the finished product QC tests. These tests are a crucial part of our quality control process, ensuring that each product meets our high standards before it reaches the customer. We perform a variety of tests, including dimensional checks, functionality assessments, and performance evaluations, to verify that everything aligns with specifications. The key point here is consistency, making sure every unit produced matches the quality and performance expectations we've set. It's also important to note that these QC tests are not just a final step, but an integral part of our overall quality assurance strategy, helping us catch any issues early. By thoroughly testing the finished products, we reinforce our commitment to delivering reliable, safe, and effective solutions. This step ties back to our overall theme of quality and continuous improvement, emphasizing that quality control is a shared responsibility at every stage of production. Now, let's talk about out-of-specification, or OOS, and deviations. So, what exactly is OOS? It refers to test results that fall outside the defined specification limits, meaning the product or material isn't meeting the required quality standards. Recognizing an OOS result is the first step in maintaining our product integrity. When we encounter an OOS result, it's crucial to follow a structured investigation process. First, we need to confirm that the sampling was correct and that our testing method performed as expected. This helps us rule out any sampling or procedural errors. Next, we repeat the test using a fresh sample to verify the result. If the OOS is confirmed, we move into root cause analysis, or RCA, to identify what caused the deviation. Finally, we document the findings and implement CAPA, corrective and preventive actions, to address the root cause and prevent recurrence. Let me give you an example to illustrate this. Suppose we find that the pH of a batch is out of specification. Our investigation might reveal that the buffer addition was not correct. Based on that, we would adjust our process recipe accordingly and retrain the operators involved. This ensures that the process is corrected and that similar issues don't happen again. Understanding and properly addressing OOS results is vital to ensuring the quality and safety of our products. and it ties directly into our overall commitment to continuous improvement. Let's now focus on documentation and best practices. When it comes to documentation, it's crucial to keep detailed records such as batch manufacturing records, which document each production run. Test logs and certificates of analysis are essential for verifying product quality. Calibration and maintenance logs help ensure equipment stays in proper working order, reducing variability. Deviations and CAPA records are vital for tracking issues and corrective actions, while stability data supports shelf life determinations. Moving on to best practices, using calibrated and qualified instruments is fundamental to maintain accuracy and reliability. Adopting a risk-based approach, as outlined in ICH Q9, helps prioritize efforts where they matter most. Regular training and competency assessments ensure staff are up to date with current procedures and standards. Finally, trend analysis of quality parameters allows us to identify potential issues early, supporting continuous improvement. All of these practices tie back to our overall goal, ensuring product quality, safety, and compliance throughout the manufacturing process. For further support, please don't hesitate to contact us. We're here to help you with any questions or additional resources you might need. And with that, I want to sincerely thank you all for your time and attention today. Your engagement is truly appreciated, and I hope you found the presentation valuable. Remember, we're always available to assist you as you move forward. Thank you once again.